Hello, hello. Do we know if the people on Zoom can hear me? Okay. Okay, everybody, we're gonna get started here. Why are you laughing? No, that's fine. This is, once people are no longer talking, I can talk loud enough, but I love that we have such a huge crowd and very enthusiastic crowd. Chuck was questioning my crowd control techniques here. I thank you for letting me know that this microphone is not working. I'm just talking to the people on Zoom so they can hear me. Okay, so it is my pleasure to introduce Elana Feldman for her master's defense seminar today. Somebody came up to me and asked me if you had to do 2 defenses because you're doing this and then you have the defense. And I said, no, you don't have to do double duty. This is just the way it goes. Okay, so I like to take these opportunities to go back and look at original correspondence that I get from potential students and also their resumes. There's some fun little nuggets, maybe just one that I will share with you. So Elana broke our, for a while we had a lot of Midwesterners in our lab, even one that is back today. I'm happy to see Jess. So Elana's from the East Coast and graduated from Middlebury College, which she got 2 degrees, environmental studies and biology. And Middlebury is home to the oldest environmental studies program in the country. So that's pretty cool. She did the RU program, RU summer program her sophomore year with Dr. Sarah Emery at University of Louisville. Some of you may be familiar with her work. Elana was nervous going into it, but she quickly discovered, she was shocked to realize that she actually had a love for plant science. So that is why she is here today. And we are glad that you found that love of plants. So she worked 5 semesters as a TA. She did a really cool senior thesis project working with a sortie model with hemlock hardwood forests in Vermont, which was one of the reasons I thought she'd be a great candidate for a lab. After college, though, she sought out projects that really melded restoration and land management with community outreach, which is something that our lab really values a lot. So she worked for 2 years as an AmeriCorps volunteer, one as an AmeriCorps volunteer in Vermont, doing all sorts of native plant restoration and outreach and coordination among lots of different entities. The TerraCorps one with Mass Audubon, she led a bunch of grassland restoration projects and conducted deer population monitoring study using pellet counts. That was very nicely worded. Pellet counts. We'll leave it at that. So she wants to pursue a career where she can really meld her love of science and restoration with community outreach. And I think the work that she's done on Great Salt Lake is a great representation of it. She had this fundamental interest in understanding the role of density and diversity and wetland plant community assembly in Great Salt Lake and Utah Lake wetlands, but did what is probably not represented as well today as all the stakeholder engagement and work with wetland managers she did as part of her project. So with that, I will turn it over to Elana. I'll take a long route and pick up my tea.
Oops. All right. Can you hear me at all with that? I don't know if the mic it's on. I don't know if it's helping you all hear me, but hopefully it is. I'm really excited to have you all here today. This is a lot more people than I was expecting. So thank you, everybody. And I'm excited to share with you what I have been doing for the last 2.5 years. So I'll give you some background to my project, then I'll go over both the chapters of my thesis, and then I'll end with some recommendations for how my research can be applied to wetland management. So I want to start by talking about biotic resistance, which is the overarching theme of my thesis, and that is the ability of a native resident community to prevent invasion by an invasive plant. And so we know that there are a lot of different factors that affect biotic resistance, and 2 factors that can very easily be manipulated by restoration practitioners in their work are density and diversity. So we know that high density is important for overcoming the mortality that naturally occurs during seeding. And we also know that both high density and high diversity are important for filling niche space. So if you have a lot of plants or a lot of different types of plants, you're going to be able to use up all the resources that are available in an area, and then the invasive plant won't be able to come in and use those resources itself. However, we do still have some knowledge gaps as to how these two factors might interact with each other, how we can use those interactions when we're designing invasion-resistant seed mixes, and then also specifically how this works within a wetland setting. So why should we care about invasion resistance in wetlands? I have a two-part answer for you. The first is that wetlands are really important. So they provide some critical ecosystem services, such as water quality, flood mitigation, and carbon sequestration. And we really need healthy wetlands if we want to have both a healthy planet and healthy human communities. But at the same time that they are so important, they also receive disproportionate impacts of human degradation. So there's degradation in all systems across the globe, but wetlands are receiving a disproportionate amount of things like pollution and draining and filling. And this extra disturbance makes them particularly prone to invasion. So it's important that we understand biotic resistance in wetlands if we want to understand how to keep these systems healthy. So my research is broadly applicable to many wetlands, but I conducted my work in wetlands specifically in the Great Salt Lake watershed, so around both the Great Salt Lake and Utah Lake. So particularly for people on Zoom who are not from this area, you've likely heard of the Great Salt Lake. It is a very large saline lake in northern Utah. You might be less familiar with Utah Lake, which is a freshwater lake located south of the Great Salt Lake. And these two lakes are very geographically close together, and they're also connected by their hydrology. Utah Lake, the water from Utah Lake flows north into the Great Salt Lake. My research is also applicable to multiple different wetland species, invasive species, but I was particularly interested in Phragmites australis, subspecies australis, which I will refer to as Phragmites for the remainder of this presentation. This species is widespread in wetlands across North America, but it first became invasive in Utah in the 1980s. And now I see it in pretty much any place there's water, really, in northern Utah. And the reason that it is such a problem is that it forms these dense monocultures that do things like reduce wildlife habitat. So you can see pretty clearly from this picture that when it enters an area, it completely takes over the area, and it makes it difficult for native plants to also grow there, which has an effect on the wildlife that are in the area and also what's happening in that community. But we also know that Phragmites can be effectively managed. So we know that things like cutting, burning, spraying, and water control, if you do them consistently for a long enough time, are going to remove the mature stands of Phragmites that are present in the area. And we also know that if we want to make sure that the Phragmites stays out of that area and doesn't return, we need to revegetate. So why is this revegetation so important? At this point, I want to make very clear that where I am seeding and where my research is taking place is not in those dense mature stands of Phragmites. It is in the places where it has been removed. So the place where I did my field experiments, both locations, it's been managed for over a decade. There's no more of those dense stands. It's now bare ground areas. But just because the Phragmites has been removed doesn't mean that it can't come back. We know that Phragmites seeds are still present in the seed bank, and they can remain present in the seed bank for many, many years after the mature Phragmites has been removed. So it'd be very easy for it to just regerminate and completely take over that area again. But even though the Phragmites seeds are in the seed bank, that doesn't necessarily mean that there are native seeds in the seed bank or that there's enough native seeds present in the seed bank to be able to outcompete the Phragmites that we know for a fact is in there. So if we did nothing, some native seeds would naturally start to enter the area and begin to regrow, but we really need to actively seed to make sure that we have enough of those native seeds present to outcompete the Phragmites. And the reason this is so important is that we need these native plants to do things like block light. So we know that Phragmites requires a very high light environment to be able to grow. And as I said, these are bare ground areas now where it has been removed. There is a lot of light hitting the ground, so it'd be easy for these seeds to germinate and take over. But if we get the native plants in there fast enough that they can take over the area and shade the ground, they're going to prevent the Phragmites from returning. So before we get into my actual experiments, I want to go over two of my treatments that I have consistently throughout my experiments. And the first is diversity. So for my experiments, I wasn't concerned so much with things like species richness or the number of species in an area. I was more interested in functional diversity, which is the diversity of characteristics that are present within a community. And so you can take groupings of species with similar ecological characteristics and put them into what are called functional groups. And these are the 5 functional groups here that I use throughout my research. So I have annual forbs, perennial forbs, rushes, bulrushes, and grasses. And you will occasionally hear me lump these into the broader terms of forbs, which is plants with large showy flowers, and graminoids, which are the more grass-like plants that aren't as showy. I also want to touch upon my seeding densities. So I have 2 densities that I use throughout. The first is a low density of 1,938 pure live seed per meter squared. And that is the typical seeding rate for wetland managers in our area. And my high density is equal to 5 times that amount. So now we can jump into my first chapter. So my first chapter focuses on 2 greenhouse experiments that I conducted. And so I will go through each of these experiments individually through their methods and results, and then come back together at the end to show the broad takeaways that we can get from both of them in combination. So for each of my experiments, I have 2 questions. The first question is always related to native plant growth. The second question is always related to biotic resistance, which I measured through Phragmites growth, or really the inability of Phragmites to grow. So the first question for my first experiment is, how does native plant growth differ by native species identity, seeding density, and Phragmites presence? The second is, how does biotic resistance differ between native species identity and density? So these were the treatments for my first experiment. I had 18 different native species that were collected from around the Great Salt Lake watershed. And I seeded one of these 18 species into each of my tubs. So they were grown in monoculture. Every tub just had 1 native species. I also had my seeding density treatment, so those 2 seeding densities that I mentioned earlier, and as well as a Phragmites presence treatment. So I had native plants growing by themselves, Phragmites growing by itself, and then some tubs that had the native plant and the Phragmites together, so we could see how growth changed when they were in competition with each other. And so any place that Phragmites was present was seeded at 3,000 pure live seed per meter squared, which is a rate that we would expect to find in managed wetlands in this area. And they were seeded at the same time as the native species. For data collection, I took weekly cover measurements, and then I also did a final biomass harvest at the end of the experiment. For my statistical analyses, I did a bunch of generalized linear mixed effects models. So to look at native plant growth, I had responses of native cover and native biomass with fixed effects of species identity, density, and Phragmites presence, and a random effect of block. And for biotic resistance, I had responses of Phragmites cover and biomass with fixed effects of species identity and density, and a random effect of block. I was also curious in incorporating a third growth metric in addition to native cover and biomass into my analyses. So to get a third growth metric, I calculated growth rates for each of my native species, and then I also determined the effect of density and Phragmites presence on those growth rates. So this made it comparable to my native cover and biomass, that I also had information from my GLMMs about how density and Phragmites presence affected those. And then I was curious about how biotic resistance might also be related to these growth rates. So I performed a quantile regression of the relationship between Phragmites cover and the growth rate. So I will start by talking about my native plant growth results, and then we'll get into biotic resistance a little bit later. So my first result is that some species grew more than others. So you can see that pretty clearly in this picture. Some of the species really took off, and some of them just really didn't. However, the effects of density and Phragmites presence differed by both species and growth metric. And so this is what I mean by that. So if we look at this one species, Epilobium ciliatum, or fringed willow herb, when we compare the low density treatment to the high density treatment, we saw no difference in cover, but there was an increase in biomass and growth rate in the high density treatment. When we compare that to, or when we look at the Phragmites absent versus the Phragmites present treatment, we saw that when Phragmites was present, cover decreased, but biomass and growth rate remained unchanged. However, if we compare that to Solidago canadensis or Canada goldenrod, which is another forb that I used, we see a different story. We see that high density resulted in more cover and growth rate, but had no effect on biomass, and Phragmites presence resulted in less cover and biomass, but no effect on growth rate. So this was a surprise to me. I expected more continuity between species in the same functional group, as well as especially between the growth metrics. But I think that this is a really important reminder that there's not going to be a one-size-fits-all approach that works in restoration for these species. All these species are going to react to different factors, and what you're measuring can even influence that. And finally, I found that density mattered less for high and low performing species than it did for species in the middle, which was also a surprise. So this is a graph of native cover. So on the x-axis, I have native species identity as a 4-letter code. On the y-axis, I have model predicted proportional native cover. And I have my densities in different colors. So my low density treatment is in blue, my high density is in red. And what you can see is that when you look at my low performing species, such as Arctic rush, we see that there's a lot of overlap in between those red dots and those blue dots. So they just didn't do that well at either density. When we look at my high performing species like beggar's tick, those dots are pretty much right next to each other. There was no effect of density. They just were going to grow well, no matter what the density was. But then when we have these middle performing species like my scratch grass, we see there's a very wide gap in between those red dots and those blue dots. So what this tells us is that some species are affected by density and really need that higher density to be able to grow. But there are other species that will just always be great or always be terrible at growing, no matter what the density is. So now I'm going to jump into my biotic resistance results. So my first finding was that some species were better at reducing Phragmites than others. So this is a graph of Phragmites reduction. So on the x-axis, I again have my species as 4-letter codes. On my y-axis, I have proportion reduction of Phragmites biomass. And I have a red line at 0 here, which represents 0% reduction. So anything that is above that line was able to reduce Phragmites. And then I again have my 2 densities as different colors. And what we see here is that in the top corner, we have 4 species in particular that reduced Phragmites by almost 100%. So there was pretty much no Phragmites that grew in those pots with those native species. I also found that Phragmites cover and biomass both decreased with the higher native seeding density, but it actually wasn't by all that much. And I again suspect that this has to do with my high performing species that again, similar to native cover, when you look at those top 4, you see there's not that much of a difference in between those red dots and those blue dots. So some species, again, might benefit at native seeding density, but high performers will be able to reduce Phragmites regardless of what density they're seeded at. And I also found that there was a negative relationship between rate of growth and Phragmites cover. So this graph is the output of my quantile regression. If you have never done a quantile regression, think about it as a linear regression. They're interpreted pretty much the exact same way. So on the x-axis, I have my growth rates. On my y-axis, I have proportional Phragmites cover. And each of those dots represents one tub. So its x location is the rate of growth of the native plant that was in that tub. The y-axis is the cover of the Phragmites that was in that tub. And you can see here, this dark blue line is representing my median data. We can see that there is a clear negative relationship between the growth rate and the Phragmites cover, suggesting that plants that grow faster are better able to reduce Phragmites cover. All right, so now I'm going to jump into my second greenhouse experiment. So my two questions here are, how does native plant growth differ by functional evenness, seeding density, and Phragmites presence? And how does biotic resistance differ by functional evenness and density? And you may be thinking to yourself, that sounds a lot like the first two questions you asked, and you would be correct. These two experiments had very similar treatments. So they had the same seeding density treatments. They had the same Phragmites presence treatments, but they differed in what was being seeded. So my first experiment was interested in species identity. So each species was growing in monoculture. My second experiment was concerned with evenness. So I took 9 of the 18 species from my first experiment, and I developed 4 different seed mixes that differed in terms of functional evenness. And this is what I mean by that. So those 9 species I had, 3 of them were from the grass functional group, 3 were from the perennial forb functional group, and 3 were from the bulrush functional group. The only difference between the mixtures was what percentage of the mixture was, or what the dominant functional group was within the mixture. So I had a grass mixture that was predominantly grasses, a forb mixture that was mostly forbs, a bulrush mixture that was mostly bulrushes, and then an even mixture that was evenly split between those 3 different functional groups. For data collection, I again did weekly cover measurements and then also had a final biomass harvest. And for statistical analysis, I again did a bunch of generalized linear mixed effects models. So for native growth, I had responses of native cover and biomass, fixed effects of functional evenness, density, and Phragmites presence. For biotic resistance, I had responses of Phragmites cover and biomass, and fixed effects of functional evenness and density. So for this experiment, I'm going to do my results from both native growth and biotic resistance together, because these are simpler than my first experiment results were. So for native plant growth, we saw that native cover and biomass both increased with seeding density. So a higher density resulted in more growth. However, we saw no effect of Phragmites presence and no biologically meaningful effect of functional evenness. So density was the only treatment that seemed to affect native growth. For biotic resistance, we saw that Phragmites cover and biomass both decreased with a higher native seeding density. However, I will mention that both densities did successfully reduce Phragmites. However, I saw no effect of functional evenness. So again, density was the only thing that seemed to matter for these results. And so this was surprising to me. I definitely expected the different mixtures to grow differently and affect Phragmites differently. And I think that the answer to that mystery lies in this graph. So this is a graph of relative abundance of the species that were present at the end of my experiment for each of my mixtures. So I have 4 panels that represent my 4 different mixtures, 2 columns per panel for my low density and my high density. On the y-axis, I have relative abundance, and then every species is represented by a different color. And so what I expected to find when I conducted this experiment was that my bulrush mixture would be mostly bulrushes, my forb mixture would be mostly forbs, etc. But you can see pretty clearly, just from looking at this graph quickly, that there are 3 main colors that just show up in all of these mixtures. And that is because really only 3 species grew and dominated each of these plots, regardless of what the treatment was within the plot. So this was surprising to me for a number of reasons. But one of them is because these species that were clearly high performing in this experiment were present in my first experiment, but were actually not high performing in that experiment. So this tells us that there's something about these three species that they really like to be in a mixture and not grown by themselves. And there's a lot of research into that that tells us why that might be true. I will also mention here that my high performing species from my first experiment were not included in this experiment. So I don't know what those ones would have done in this situation. But something that I also thought was interesting from these results is that you may remember from a couple slides ago, the only significant results that I had were related to density. I again suspect that that has nothing to do with seeding a mixture at a high density, and specifically in seeding these species at a high density, because two of these three species were significantly affected by density in my first experiment. So again, it seems to just be the species driving all the results and not actually using different mixtures. So now, if we take both of those experiments that I conducted in the greenhouse, combine them together, what do we learn? So first, species identity seems to drive final, native, and Phragmites cover and biomass. So we saw in my first experiment that there were certain high-performing species that had a lot of control over the final native biomass, as well as the Phragmites cover and biomass. And then we saw in my second experiment that the mixtures didn't really seem to matter. If you had high-performing species present in those mixtures, those were what were going to drive the results. Second, I found that Phragmites presence really depends on the species and the growth metric that you're looking at. So I have no blanket statements about how competing with Phragmites in these revegetation efforts affects a native species as a whole. It seems to depend on what species you're measuring and what growth metric you're using for that. And then I also found that a higher native seeding density both increased the native growth and decreased the Phragmites growth. But again, the importance of density was species specific. So we saw in my first experiment that not all species reacted the same to the high density. And in fact, some of these high performing species don't need to be seeded at a high density to produce the same results. Okay, so that wraps up my first chapter. Now we can get into my second chapter. So my first chapter was focused on my greenhouse experiments. My second chapter focused on my field experiments. And so for these experiments, I again had two questions. How does native plant growth differ by functional group identity and seeding density? And then how does biotic resistance differ by functional group identity and seeding density? So these treatments were again somewhat similar to my greenhouse treatments, but also a little bit different. So it had the same native seeding density treatment, so that exact same low and high density that I used for my other two experiments. But what I was seeding this time was more focused on functional groups than it was on the evenness or the species identity. So you may remember this slide from the beginning of the presentation. These are the 5 functional groups of interest throughout my research. And for this experiment, I had 5 different mixtures, each with 3 species. And every species in the mixture represented just that one functional group of interest. So I had an annual forb mixture, a perennial forb mixture, a rush mixture, etc. So they weren't being mixed together. Every mixture was just one of these groups. And my third treatment was location, although admittedly this was not a treatment that was included in any of my statistical analyses, but I conducted the same experiment at 2 different locations to see if there'd be a difference in the effects when you planted in different places. So one of my experiments took place in Farmington Bay, which is a bay of the Great Salt Lake. The other took place on the north shore of Utah Lake. And so, as I said earlier, these 2 lakes are geographically close together. They also share a lot of the same plant communities. Utah Lake flows north into the Great Salt Lake. They're very closely connected. However, there are some differences between these 2 lakes, including how they're managed. So for example, Farmington Bay is very heavily managed. They have complete control over the water that's coming into the bay. So I suspected that because there are these types of differences between these 2 locations, there might be differences in the results of a restoration effort. For data collection, I collected cover bi-weekly. So every week I was at one of the 2 locations. And I also collected information on water depth. So at the block level, I had groundwater wells that went about 2 meters deep that gave me a sense of where the water was in relation to the ground. So again, for my statistical analyses, I did a series of generalized linear mixed effects models. In these models, with one difference being that I paired each of these models with a Dunnett's test so I could get a better sense of how each of the treatments related to the control. I had fixed effects of functional group and density and a random effect of block. And for my responses, I had 7 different responses this time. So I had total native cover. This included everything I seeded, plus any native plants that I hadn't seeded, but that came up from the seed bank or had blown in. And then I also had total invasive cover. But in addition, I had 5 different responses related to my 5 functional groups, so I could get a sense of any of these functional groups reacted differently to just the total cover. So to get into my results, my first result was that seeding had very little effect on cover. So after 2 years of monitoring these plots, I found very few significant effects or any differences between treatments or between the treatments and the control. And this is a picture from the end of my first sampling year. So this was taken on the last day of sampling in 2022. You can see that there are not a lot of things growing. There's really no difference in any of these plots to how they look. And so I did not test this directly, but I suspect that the reason for this has to do with water depth. So these are my water depth graphs. On the x-axis, I have date. On the y-axis, I have the depth. The top panel is Farmington Bay. The bottom panel is Utah Lake. And I have a red line on 0, which represents ground level. So any dots that are above this line means there was standing water in the plots. Any dots that are below this line means the water was drawn down into the ground. And you can see from both of these pictures that the dots are pretty far below that red line. So if you were living in Utah in 2022, you remember we were in a very severe drought. I was theoretically planting in a wetland, but this is what all of my plots looked like. This was very different from 2023, where we had prolonged flooding. So you can see from this bottom graph of Farmington Bay in 2023, I had standing water in all of my plots. In fact, it was about a foot of standing water in all of my plots for almost the entire growing season. So a very different type of severe weather event, but severe weather events in both years. You may be wondering why I don't have a 4th graph of Utah Lake from 2023. And the reason for that is that the lake was so flooded that I could not access my plots for the entire season. So I only have 1 year of data from Utah Lake. So again, 2 very different events, flooding and drought, but plants don't like to grow in a wetland under either of those conditions, unfortunately. My second finding was that invasive species really took over the plot in the 2nd year. So again, I only had 1 year of data from Utah Lake, but at Farmington Bay, I had 2 years of data. The first year cover was low, but it was almost entirely native plants. My 2nd year cover was still low, but it was, those native plants were pretty much gone. It was almost entirely invasive plants. So this is a picture of one of my few plots that did have high cover. And you can see that it is 100% Phragmites and invasive cattail. So that was very sad, but it does make a lot of sense. Invasive plants can survive a broad range of environmental conditions, which is part of why they are invasive. So it makes sense that the invasive plants would be able to survive that drought and continue to survive during the flood, even though the native plants wouldn't be able to do that. But on a happy note, I was able to find 3 native species that seem to do well, even with the extreme weather. The first 2, saltgrass and alkali bulrush, were coming up naturally in the seed bank and really did well both years. Both of these species are already being used in restoration projects in the area. So that's a good sign that these species can survive that. And the third one, hard stem bulrush, was a species that I saw from my seeding efforts. It was the only species out of everything I planted that had any significant effects at all. And it did well during the drought, and then it continued to do even better during the flooding. So this is a great potential restoration species that I would encourage to include in restoration seed mixes. And my third finding was that plant communities did differ between the two locations that I was looking at. So this is a picture from Farmington Bay at the top and Utah Lake at the bottom. And these pictures were taken on the same day. So you can see that at the end of my first growing season, Utah Lake had higher cover, it had more species, it also had higher diversity than Farmington Bay had. And I did not test this, but I suspect that this has to do with the differences in salinity at the two locations. So the top is from a saline lake, the bottom from a freshwater lake. And even though the two plant communities are pretty similar at the two lakes, we can see that that still has an effect on plant community outcomes. Okay, so in summary from my two different field experiments, first, I found that water depth can overwhelm the effects of seed treatments. And what I mean by that is it doesn't really matter what you seed. If there's not enough water for the plants to grow, or if there's too much water for the plants to grow in, your mixtures are, what you have in your mixture is not going to be important. However, I did find that some native species are resilient to extreme weather. And this is very good news because we know that the weather is only going to continue to become more extreme as the climate continues to change. So it's good that we can start to identify these species that can survive in this range of conditions. And finally, I found that environmental differences at each site can affect plant community assembly. This is not news to managers, but it's an important reminder that even if you have locations that are geographically very close together and that tend to have the same plants growing at them, doesn't mean your restoration efforts are going to look the same in both places. Okay, so now that we've gone over both of my chapters, if we consider all of my experiments together and think about how this actually affects wetland restoration, these are 4 recommendations that I have for managers. So the first is just a reminder to consider species under different contexts. So we saw in my first chapter that different species are going to react differently when they're in mixtures, when they're at a high density, when there's Phragmites present. And we saw in my second chapter that environmental conditions can really affect what species are growing. So this is just a broad reminder that we can't always predict what these species are going to do in every single scenario because they will react differently under different scenarios. However, I do recommend including both high-performing and resilient species into seed mixes. So we saw from my first chapter that there are certain high-performing species that are just really good at growing, really good at preventing Phragmites from growing. So if you're trying to design an invasion-resistant seed mix, I highly recommend including those species. However, we saw in my second chapter that only some species are able to survive a broad range of conditions. Because we never know what nature is going to throw at us, it's important to include resilient species to try to make sure that something is living no matter what you plant. I also recommend selecting a seeding density based on the species that you are seeding. So we saw again from my first chapter that some species really do better when they're seeded at a high density, but some species don't need that high density. They perform the same at the low and the high density. So there's no reason to spend 5 times the amount of money on 5 times the amount of seed if it's not going to make that much of a difference. Ooh. And finally, I recommend considering the environmental differences because you can't predict that a seed mix is going to react the same no matter where you plant it because it will be affected by differences at different sites. Okay, so with that, I have a lot of people that I need to thank in addition to all of my funders, which are listed here on the slide. First, I would like to thank Karin, my advisor. I really could not have asked for a better advisor, and I feel so lucky to have been part of this lab. I'd like to thank my committee members, Kari Veblen and Tim Walsworth. You both have given me a lot of really valuable help throughout this process. And you pushed me to do take a few extra looks and a few extra statistical tests on my research, which is, it was good. It was good for me to do that. I would also like to thank several other people who made this research possible. So first I want to thank Alec Hay and his staff at the USU greenhouses, who are some of the most patient human beings I've ever met, and really saved me so many times when I was on the brink of collapse. So I really appreciate everything they've done to help me. I'd like to thank Susan Durham, who helps me a lot with my statistical analyses and in interpreting my statistics. That was a major help. Thank you. I'd like to thank David England and Keith Hambrecht, who are two wetland managers who allowed me to do my field experiments at the sites that they manage. So they both gave me access. They both helped me a ton with logistics, and they both have just spent a lot of time in the field with me, teaching me things about management. So I really appreciate all the time that they have spent with me and how much they've taught me over the last 2 years. And I'd also like to thank Shane Sterner, who helps the lab with seed collection and seed cleaning. So a lot of these experiments literally would not be possible without him. So we're very happy to have his help. I would also like to thank the numerous members of the Whirl Lab who have helped me over the last 2.5 years. It really is, it's much more of a family than it is a lab group. And I am so happy to have had all of these people in my life. It has been wonderful to get to know everyone. And I'd also like to thank my friends and family who gave me continued emotional support and answered many late night text messages, even though they had no idea what I was talking about, but they were willing to try to help me anyway. And I really appreciate that. And with that, I can take any questions. Yeah, Phaedra.
I was just curious if in the first set of experiments in the laboratory or the greenhouse, excuse me, that if you delved further into what the mechanism might be by which those densities affected the treatments.
Yeah, it seems like it's just some species, like the high performing species that were not affected by density. It seemed like they just grew these very broad leaves. So even if there was just a couple of stems that were there, they were still taking over the plot and completely shading the area. So the Phragmites still wasn't able to grow, and there was still a good amount of biomass. Whereas some of these other species were a little bit more spindly. So we really needed that high seeding density to actually get them to cover the area. That's a major thing that comes up when you're comparing like grasses to forbs. A lot of these grasses, you need the high density because they're sticking straight into the air, so they're not actually going to produce very much cover. But some of those forbs, one leaf will take over half the plot. So, yeah, Chuck.
This is a bit of a follow-on to Phaedra's. You didn't see a functional group signal or much of one. Is that because the functional group classification just wasn't particularly appropriate for this particular question? And on hindsight, could you parse those species into different types of functional groups?
Yeah, I knew you were going to ask me that. So I think that's a really good question. So I think I do think part of the lack of effect was just the high performing species, but I do think part of it is that my functional groups weren't specific enough. And that is something that comes up a lot just in plant science in general. The functional groups are not nearly as well defined as they are in terms of like macroinvertebrates. So I do think that in the future, the functional groups could incorporate, I incorporated a few different characteristics, but we could incorporate even more, and we can incorporate things like seedling characteristics. That's another major problem that comes up a lot in plant science. We use these adult characteristics, but they're not competing as adults, they're competing as seedlings. So those functional groups might not be consistent from the seedling stage to the adult stage. So I think that's a good point.
I'd like to know how you worked around this missing panel. You measured it, or
I
you
meant
came
to
in
bring
and
that in
edited
my
it?
introduction. How did Elana handle this with help from a friend? But I understand, you explained it.
It's all Megan. I have Megan to thank for this.
We made a box on the slide that was the size of the hole in the screen. make sure it's not there. Precise calculations. Teamwork. The recipes for success for Elana Feldman. Any other questions? Oh,
Ask a
here.
question if you want. I just had a question, in terms of your management recommendations. So you were recommending, obviously, maybe more use of those high-performing species, those native species. And I guess I just am wondering if there's any research in terms of those species in general, of if you are kind of focusing on those, and they're, you know, obviously doing really well, does that foster further like native species assemblage, or does that kind of limit the assembly in the future of like diversity, or is it easy to plant into those stands that you've already planted?
Yeah, that's a really good question. This is something Karen and I have talked about, whereas we don't want to encourage people to only plant those high performing species, because something that I didn't really mention here, but a lot of those high performing species are considered weedy they're all native, but they are considered weedy by a lot of managers because they do grow so well. So we don't want those to be the only thing that's present in the area. So we're still encouraging very diverse seed mixes. Some of those high performing species, so we know that something is going to do really well, and then the lower performing species, so that we can continue to encourage diversity in these seed mixes. And no, I have not actually seen any research into those species specifically, besides what we've done in the lab. Definitely in the greenhouse, they will completely take over and kill some of the other native plants, which is unfortunate. We have not seen that happen in the field. But I do think that's something that, I don't have any particular concerns about any of those species, but we should continue using very diverse seed mixes to try to avoid something like that happening.
Cool, thanks.
So obviously water depth played a pretty big role in like how these field experiments, like how you saw reseeding occurring. Do you have any insight on like what an ideal like growing scenario would have been for some of these species? Like what is a water depth that would have been appropriate?
Yeah, Maddie's doing research into that actually. So we have other people in the lab who are looking into that. In general, we would like some amount of standing water at the start of the season, maybe not standing water at the end of the season, but have it still be moist. And yeah, it was just sort of unfortunate that for my 2 years, I had like very severe drought and very, very prolonged flooding. We do know, so luckily at places like Farmington Bay, they have a lot of control over what water goes into what area. So managers do have some level of control over how wet the ground will be. That didn't really occur in the areas necessarily where I was seeding, and they can't do anything about the extreme weather. But yeah, we would like to see, I don't know, moist soils, and luckily there are managers who can do that and make sure that that happens in the wetlands.
I'm curious, your field experiments, did you just seed the first year? Obviously, would you recommend that, or would you recommend people try and secure funding for multiple years of seeding?
Maddie's also looking at
I'll ask her.
but yeah, I think in general it's good to seed multiple times, because you don't know what's going to happen in any of those years. That's definitely a major limitation to restoration, because seeds are very expensive. So it's really hard to buy seeds for multiple years in a row, but that is sort of going to be your best shot at making sure that something grows. Yeah, I would generally recommend seeding multiple times, but yeah, I did not do that.
We have a Zoom question, which is from Grace Shaw Jane. I noticed on the biotic resistance plot that there are species that seem to facilitate Phragmites growth. Are there any ideas as to how or why that happens?
Yeah, thank you for coming, Grace. So I think yeah, that was interesting and not something I expected. And I would say those species where it seemed like Phragmites grew more, those were all species in which none of the native plants grew. So I actually ended up not including those in some of my statistical analyses. Yeah, I don't know why that happened, besides the fact that there was just no competition at all. But I don't know why there would have been more Phragmites there than it was. in the control plots.
We have one from Keith as well. Keith, do you want to unmute?
Hi, Lana. Great job. Great presentation. So my question, of course, is management related. Now that you've gotten to know these two sites, maybe we'll just focus on one of them, Farmington Bay. If you were going to continue with revegetation at the Farmington Bay site, knowing what you know now, what species would you choose, and what would your approach be for this following season?
That is a good question. Thank you, Keith. I'm happy to say a lot of the species that I would continue to encourage are species that are already being seeded there. So it's the alkali bulrush, the saltgrass, and the hard stem bulrush that I know have already been added there. Those are the ones that were my resilient species and that did well. However, I would encourage seeding more forbs. So the beggar's tick, sunflower, I'm trying to think of I think the Epilobium ciliatum, the fringed willow herb, those all did really well. That's something that I've not seen included in the restoration seed mixes just when I've been helping out on restoration projects. And I think that those do have a lot of potential. Obviously, we saw that I couldn't get those to grow as well in the field, so there are some limitations to those. But I do think, Rumex maritimus also, that I think also has a lot of potential to do really well in the field. So I think it will just sort of be a lot of what managers are already doing, continued seeding efforts, continuing to manage water to try to ensure that they're getting enough water to be able to actually grow. And then just the continued management of the invasive plants to reduce that, that competition pressure.
We have one from Eric Nicoli as well.
Okay, thank you for allowing me to unmute. Great job on this, and it's great to see some good follow-up on a lot of work that's been done over the years. I noticed some of these species are really hard to come by the seeds. Joe Pye weed, Canada goldenrod, and a lot of the forbs are difficult to find. Is there any effort to increase the availability of some of these forbs, especially to use in these type of things, or is it mostly still just going to be field collection? Thanks.
That is also a great question. That is also something that the lab is working on, luckily. So the lab has some projects interviewing managers and vendors to try to get a sense of what the demand is and why vendors are not providing those seeds. So as a lab, we do collect our own seeds in the field. So we were not purchasing these. We did that for a variety of reasons. But so I actually don't know if there are any places in this area that are selling those seeds besides Shane, who helped us collect some of those. But yeah, I think that's a major problem for restoration. Something we're trying to encourage as a lab for vendors to consider these other restoration species. But yeah, I think that's definitely currently a major limitation and hopefully will not continue to be, but likely will be for a few more years at least.
Thumbs up from Eric.
Yeah, Rachel?
What's the story behind the hazmat suits?
That is me and Sam. So we had to go out and Margaret assisted, of course. We had to go out and spray some of the invasive plants in my plots because since I was growing native plants, they weren't able to manage that area. Then you guys saw in my photos earlier that the Phragmites really took off this past year with the flooding. So we offered to spray some of the invasives in and around my plots for the managers to help them. And we didn't want to inhale anything, so we really got all decked out. So it was a day.
And if there aren't any more questions, we have one last one from Austin on Zoom.
Oh, no.
What was the significance of Raymond's support during this process? And do you think you would be proud of the quality of your work?
Thank you, Austin, for that important question. For anyone who doesn't know, Raymond is my pet rabbit. He actually, I was gonna say he provided a lot of emotional support, but the truth is that he did not. He is the king of the household, and he is upset. He actually stomped his foot at me yesterday while I was practicing my presentation because I woke him up from sleep. Raymond just continued to demand food and love. I can't say that he helps with research specifically.
Okay. On that note, let's thank Elana for excellent presentation.